All right, I'm just going to review some of the things from the online activities. And the one thing is that the cell is the basic unit of structure. And I think I talked about last time that Western Technical College, think of it as a human being, the whole organ system, organism, made up of organ systems. And the people that work at Western are the actual cell. It's the unit that is doing the work, right? Doing the work of the cell, so of the organism. So we are made up of a number of cells, each with a specific function. So when we say that the cell is the basic unit of structure and function, it just means we are made up of cells. So if we break us into the smallest pieces, it's cells. So our skin is a layer of cells, or our heart is a layer of cells, broken up into tissues, right? But it's cells. Everything is made up of cells, so it's the basic unit of structure. And then function, all the organs, what they do is based on what the cells that they're made of, what their function is. So the basic unit of structure and function is the cell. Now your online activity, and in lab this week, we talked about the different parts of the cell, so I'm not going to review that. You can read that. But what I do want you to be aware of is this internal environment. When we say that cells are part of the internal environment, it just means where all this physiology is happening is in the internal environment. So when we talked about feedback systems last time, we talked about how blood sugars drop, the cells of the pancreas detect the low blood sugar, and the cells release glucagon to raise the blood sugar in between meals. So that's a feedback response. And the cells are communicating with this internal environment and detecting changes to see if they need to kick in and do something. So the internal environment is made up of a couple of different parts, and this is what I want to talk about. So the internal environment is made up of the interstitial fluid. Interstitial fluid is a term that you're going to need to know, and we're going to talk about it a lot this semester, because this is the environment where cells are interacting. So the interstitial fluid is the fluid between the cells. Plasma is the fluid that is in our blood vessels. Some people donate plasma for income, right? Because there's stuff dissolved in the plasma that's essential for life for some people that lack those things in their own plasma. For example, clotting factors. So if you're someone with hemophilia, you lack the clotting factors in your plasma, so you use donated plasma. They give that person clotting factors from donated plasma so they don't bleed out if they cut themselves. Yes? But anyway, when we talk about the internal environment, we're talking about the fluid outside of cells and the fluid inside of cells. So outside of cells, we call that extracellular fluid. So any fluid, whether it's in the blood vessels or just between cells hanging out in a tissue, that is called extracellular fluid. We can abbreviate it ECF. It means fluid outside of cells. So that's one part of the internal environment. The other part of the internal environment is fluid inside cells. We call that intracellular. So if you look at this bar graph here, the tall blue bar is fluid inside cells, intracellular fluid. So when we talk about the body being made up of water, most of that fluid we're talking about of the internal environment is water. Where is most of the fluid found? Inside cells, right? So if we become severely dehydrated, first what happens is we lose fluid from our tissues. Then we lose it from our blood vessels, the plasma. And the last resort is losing the fluid from the cells themselves. So when a mother brings in her baby who's had vomiting, diarrhea, and wouldn't take anything by mouth, and they're listless and just laying there almost comatose, you know that they are not only dehydrated from the interstitial fluid, but also the plasma, and also the cells are shrinking because they're not functioning properly. The nerve cells, the cells that make up our brain and spinal cord, they need a nice amount of fluid to keep them plump and operating correctly. So we need to keep ourselves hydrated if we want optimal brain function. So is it a good idea to have a glass of wine at night, right? So that causes you to lose fluid. That's somewhat of a diuretic. Get up in the morning, have a cup of coffee, which is also a diuretic, causes you to urinate, and then come to class and expect to operate at a high level? No, because we're not hydrated. We wake up dehydrated. Do you feel thirsty in the morning when you get up? You should. Everybody is a little dehydrated in the morning. So the best thing you can do is pour yourself a nice big glass of water, plump up those cells in your body, keep yourself hydrated, and you're going to function better in your classes or at work or wherever because your cells are hydrated. When we get thirsty, that's a sign of dehydration. We should never feel thirsty. If we're properly hydrating ourselves throughout the day, you should never feel thirsty where your mouth is sticky. Because what does that mean? Where did you lose fluid if you have a sticky mouth? Which fluid compartment of the three shown here? We've got inside cells, that's intracellular, interstitial, or plasma. If you feel sticky in the mouth, interstitial, yeah, it's the fluid between our cells, the fluid in the tissues. We lose fluid there first because that doesn't affect the body, right? Just makes us feel uncomfortable. But then if we lose enough fluid in our tissues, then we got to pull from the plasma. What happens to blood pressure then if we're losing plasma? It goes down. So that's a sign of dehydration, isn't it? How do you think the heart would respond if their blood pressure is lowering? Would it speed up or slow down to keep blood flow to the tissues? We've got to keep our blood flow to the tissues. It's going to speed up. So that's a sign of dehydration, is a rapid heartbeat, right? That's a negative feedback mechanism. And then lastly, again, if you're lethargic and sleepy, then you know you're really dehydrated. So we usually stop. Our body kicks in. If you're normally healthy, our body kicks in after we lose some from the interstitial fluid. You feel a little dry, sticky, you drink some water, you're better. So it's really a good idea to drag a water bottle around with you. It really is. And don't buy water. There's so many cheap water bottles you can buy. And on campus here down right outside the bookstore, we have a thing that fills water bottles very quickly. So I make it a habit when I come in to class, when I have my water bottle. Now, if you have 4 kids that all want to bring a water bottle to sports and school, at my house, I can never find a water bottle. I probably had 10 and they're all gone. We even had that battle this morning. Who took the last water bottle? What?
Mine in my room.
Do you?
Yeah,
I need a safe in my room because my kids are even in my room pilfering around looking for things.
This has my school on it. You can't.
Oh, I like that. I like that. Yeah. Not your school. I should. But then, see, in my office, I don't have access to a water thing in my office. So by bringing it with me, I fill it on my way in and I fill it on my way out. Anyway, it's a good idea to bring water with you, especially in healthcare. We're running around so much, we don't have access to water as easily. Hey, bring a water bottle. So the 3 compartments, know what they are: interstitial, between cells, intracellular, inside cells, and plasma is in the blood vessels between the red and white blood cells. So the red and white blood cells are the solid parts of our blood, and that straw-colored liquid is the plasma. That's the other fluid. All right, so then we're going to kind of just scroll through this here and talk about movement across the membrane. There's 2 types of channels in the membrane. There's gated channels and non-gated channels, which means things cannot just pass in and out of our cells unless we need it, unless we need that substance. We can't just let any old thing in and out because it would drain out. For one, we would lose important substances that we might need in our cells, like glucose. We don't want glucose to just leak out of our cells randomly. So we have some channels that are gated that prevent the loss of glucose. Some are non-gated, which means they can just freely come through. So what drives the movement across a non-gated channel is concentration differences. For example, if I have a lot of sodium in my diet, I absorb it into my cell, lining my digestive tract. Then it goes into the blood that are found, the blood vessels that are found around the digestive tract, in the plasma. So I've got all this sodium. Let's say I went to McDonald's, had a high salt meal. I have all the sodium in my blood. It's going to pass out of the plasma into the interstitial fluid. And then what? How do you feel a couple hours, maybe the next day after eating a lot of salt? Bloated, a lot of water. Yeah. And you feel dry because salt pulls water out of when salt enters a space, it draws water in. Because water flows from dilute areas to more concentrated areas. So anytime we have a salty region, that pulls water into that region. So now you have all the salt in your tissues, and then that salt goes into your cells. But only if there's more salt outside the cell compared to the inside of the cell. So we have these concentration differences that determine what direction things are going to move. Sugar always goes into cells. And there's a kind of a detailed explanation for that, but we're not going to talk about that in this class. It's an advanced A&P concept. But we need insulin to open up the channel to allow glucose in. And we also have other ways that glucose enters through proteins, but they are gated channels, okay, gated. So there's two types of gated channels, which means they need something to open them. One is called a ligand gated. Ligand means some chemical has to bind to it. So we're going to discover this semester that some of our cells don't let in sodium unless a chemical like acetylcholine binds to that channel. Once it binds to that channel, it opens up and sodium can enter. Voltage gated means there has to be some change in electrolytes on either side of the membrane that cause a channel to open. And when we talk about the action potential later on in the semester, that's an example of a process that requires voltage gated channels. But I don't expect you to understand that right now. All you need to know for right now is there's two general types of channels. There's non-gated, where things can freely flow through depending on the concentration on either side of the membrane. And then gated channels that can be either ligand gated, meaning a chemical has to bind, or voltage gated, that there has to be some charge, some voltage charge on either side of the membrane, some change that triggers those to open and close. So here's an example. What type of, well, you tell me, what type of channel is this showing? It's a gated channel. Which type of gated? Is it voltage gated or ligand gated? Ligand gated. And in this case, what is the ligand? Yeah, the acetylcholine. That has to bind, then it opens up that channel and allows that sodium to enter. So we say that our cell membrane is selectively permeable. Permeable means you can pass through. Selectively means only certain things can pass through. Not everything can come through, only certain things depending on what the needs of the cell are. So some of these, some ways that things get across the membrane that can't dissolve right through the membrane, sometimes they need an enzyme to break them up into smaller pieces, and then they're carried across the membrane. They're not necessarily a channel, but it's a protein that binds, changes its shape, and dumps it on the other side. So this is another type of transport. It's just called a carrier protein. So when you look at all the different types put together of how things get from one side of the membrane to the other, there's diffusion, osmosis, filtration, and specialized transport. So we'll talk about a couple of these, but the key thing that determines how they're going to get across is what type of substance am I transporting? Can it dissolve freely, or does it need a helper? So some things that can dissolve in the membrane are those that are fat soluble, because what makes up that plasma membrane? What are those little circles here, the ball with the two tails? What are these called? Phospholipids, right? You should know that from the worksheet you filled out for today. I need to stamp those before we leave. So yeah, so the phospholipids, which is a lipid, it's a fat, it's an oil. So water and oil don't mix, right? We know that, like, if you have salad dressing, vinegar and oil, you have to shake that bottle really well, or put them on separately. Either way, if we have a substance that can dissolve in water, then it's not going to be able to dissolve in the membrane. So things like sugar, water, glucose, ions, they cannot dissolve in the membrane. They have to be transported across. So they need something to carry them across. Things that can dissolve in alcohol, in oil, is like alcohol, carbon dioxide, and oxygen. They can dissolve right through the membrane. Another fatty substance like cholesterol, steroid hormones like estrogen, testosterone, progesterone, those are all fatty molecules. They can dissolve right through the membrane. They don't need a helper. So if they can dissolve without a helper, we call the process of just dissolving in the membrane and going to the other side, we call that diffusion. So diffusion is anything that can just dissolve very easily through the membrane without any help. Or what if we have a non-gated channel? Could it diffuse through there just as easily? Yeah, so that's another example. Some of these water-soluble substances can go through if it's a non-gated channel, and we do have some of these channels. We call them leak channels. So some of most of our cells are leaky to sodium. So sodium has has an assortment of channels. Some are gated, some are leaky. So if it's open on both sides, as long as there's a higher concentration on one side compared to the other, sodium is going to push through that non-gated channel. So if it's fatty, it doesn't need a channel at all. It can just dissolve right through the membrane. So if it's, like I said, if it's alcohol, carbon dioxide, oxygen, or some fatty substance, it can dissolve through the membrane. If it's not, if it's water soluble, it needs a non-gated channel to get across. If we don't want to use any energy, that is. So when we say things flow downhill, it just means from higher to lower concentration. Missing an R there. And then viscosity tells us how thick that liquid is. If something has a high viscosity, it means it's really thick, like molasses. If it's really thin, low viscosity, it means it's very watery. So which do you think diffuses more easily? A thick liquid or a watery, thin liquid? Watery and thin, yeah, for sure. So if I'm dehydrated and I have thicker blood, I have higher viscosity blood, things moving in and out of the plasma, it's a little bit more difficult, isn't it? Because it's thicker, there's less fluid there. So maintaining a low viscosity in our tissues and in our blood is important for things to diffuse in and out of our cells properly. So again, with diffusion does not require energy, no ATP, that's the energy molecule for our cells. Things can diffuse just easily from higher concentration to lesser concentration. The key word on this slide is water, movement of water. Water flows from where there's more water to where there's less water. So if I have a sugary solution and I have just a plain old water solution, which direction is water going to flow? From the pure water to the sugary, or from the sugary to the pure water? Pure water to the sugary. Where is there more water concentration? In the pure water, right? So water always flows from a dilute to a more concentrated solution. Because osmosis is what we're talking about. We're talking about the flow of water only. We're not talking about the sugar solution, because which way would the sugar dissolve or diffuse? It would diffuse into the water because there's more sugar on one side of the membrane than the other. So there's a couple of terms we use to describe a solution. Isotonic means that that solution is the same as the solution around it. So if a cell is isotonic, that means the concentration of solutes in water is the same as the interstitial fluid. And isn't that what we would want to maintain if we're just hanging out in homeostasis? There's not really a net movement of water or solutes in any one direction. We're just kind of hanging out. So when you go into the hospital and you're not feeling well, the first thing that they do almost to everybody who comes in the ER is and you're going to be admitted to the hospital, they're going to put you on some IV. They're going to give you 0.9% sodium chloride because that's isotonic with our cellular and interstitial fluid. So they're going to put you on that just basic IV fluid. So it doesn't mean anything ominous or scary when all of a sudden you see a bag hanging there. It's just they want to maintain your fluid levels, and they might be adding some medicine, so they want to have just a nice, maintenance fluid up there. Hypertonic means it's a concentrated solution. So wherever this is, if it's a cell in a hypertonic solution, it means we're putting the cell in a solution that is more concentrated. So what is that going to do to the water inside the cell? It's going to draw the water out, yep, and then the cell is going to shrink or crenate. We call that. When a cell shrinks, we say it crenates. So we don't want a hypertonic solution around our cells. We want to keep it isotonic. And then hypotonic is when you put would be when the cell is in a low solute concentration, a very dilute solution. That's going to cause water to flow into the cell because now the water is more concentrated than the environment around it, and water is going to flow into the cell and cause it to burst. So lysis is when something blows up and explodes, breaks down. So that would be a cell in a hypotonic solution. So high water content, if that's what's in the solution, we call it a hypotonic. Low water means hypertonic. And if it's the same, then it's isotonic. This movement, though, across the membrane does not require energy. So movement of water does not require energy. movement of solutes through a leak channel or dissolving through the membrane does not require ATP energy. Another type of movement is called filtration. Filtration is when there's a pressure difference. So in the kidneys, blood pressure is adjusted to make sure we have enough flow of blood into our kidneys to filter out the plasma and get rid of waste. So that's a special type of movement from one compartment to another, and that's called filtration. So pressure differences is what drives that. So that's not an energy ATP requiring process either. It's just blood pressure from one side of the vessel to the other. No ATP for that either. Facilitated diffusion is when that substance doesn't dissolve in the membrane, it's water soluble, and it needs a carrier. So it needs a carrier to go across. It's going to change its shape, but it doesn't require energy to change its shape. It's just a chemical reaction that occurs. That carrier changes its shape and brings that across the membrane. But where is there a higher concentration in this example? Outside the cell compared to inside the cell. And that substance is moving from higher concentration to lesser concentration. So in this example here, the substance is moving down the concentration from high to low. No energy to go from high concentration to low concentration with a protein carrier. Yes?
The protein carrier is specific to certain molecules?
Yes, definitely. Yep.
So once it moves one molecule, is that often it's like done until?
Well, then it goes back to its original shape until then another one will bind, change its shape. Yep, and it'll keep transporting until the concentrations are equal, or it'll just keep moving it across. If we keep sucking it up, like in the case of glucose, we're always using the glucose, so that keeps driving it into the cell. And some glucose does move by facilitated diffusion. And glucose moves by a number of ways, actually. Now, the next couple are all examples of types of movement that do require energy. In this case, the sodium-potassium pump, how many sodium are bound to this carrier? 3. And how many potassium? 2. So in one cycle, we're moving 3 sodium and 2 potassium. And if you look at the arrows, which way is potassium moving? Out of the cell. And sodium? No, potassium is moving not out of the cell. Look at the arrows. Into the cell, yeah. Sodium is moving out of the cell. Now, if I look at concentration differences, if you watch the animation, which we already showed in the online activity, you'd see that sodium is constantly being dumped out of the cell, even though there's less sodium in there. So in this example, the sodium-potassium pump, those ions, sodium and potassium, are moving opposite to their concentration gradient. So we are always going to have more sodium outside the cell and more potassium inside the cell because this pump is pumping them against their concentration gradient. And it's necessary to do that when we study the nervous system and talk about flow of ions and how that relates to action potentials. We'll get to that. But for right now, just know that they're moving from an area of lower concentration to higher concentration. So that requires energy. And here's the example I use for that. If I have a leaky boat and I sit out in my canoe, I have a leaky canoe and I'm sitting out in my canoe, where is there more water? Outside the canoe? I'm in a lake. There's more water outside the canoe or inside the canoe? Outside. So water is going to flow in through those leaks. Does it require any energy on my part for that water to come into the boat? No, it's going from higher concentration of water to lesser concentration of water. No energy required. Now, if I want to take care of my boat and not sink, I need to put it from less concentration to higher concentration with a pail. Does that require energy for me to bail the water out? Yes. So think of it that way in terms of transport. If I'm going from lesser concentration to higher concentration, requires energy, requires ATP. Think of it downhill from high to low. I'm on a slide. Does it take energy for me to go down the slide? High to low? No. Does it take energy for me to go up the slide? Yes. So think of it that way too, to keep the two straight. Because there'll be a couple of questions on the exam talking about concentration differences, which require ATP energy, which do not. Okay, secondary active transport is where some substance is riding along another substance's concentration gradient. So here's the pump, and what do we say it does? What does it build up outside the cell, the sodium-potassium pump right here, the orange one? Yeah, it builds up sodium outside the cell. So when sodium comes to this carrier molecule, this is facilitated diffusion, it's going to bind to sodium, change its shape, and dump it on the other side from high to low concentration. So does that require energy to go from high to low? No. Glucose, though, is going to ride low to high. See how there's less out here and there's more here? So that requires energy normally, correct? But it's going to ride in on the conformation, the molecular shape change of this protein carrier, which will transport it inside the cell. So we call it secondary active transport because this didn't provide the energy, this did. So it's kind of like a taxi cab, right? Or a free ride on a taxi cab because sodium put in the energy using ATP to build up outside the cell, and then it comes in freely into the cell through this carrier molecule from high to low, but glucose is riding along with it.
So
That's called secondary active transport.
sodium is paying for the cab,
Exactly,
and the glucose is just riding along.
exactly.
Okay.
Yep, yep. Okay, so sodium paid over here to get the energy to build up outside the molecule, then it just diffuses in through that carrier molecule, and glucose comes along for the ride against its concentration gradient. Okay, and then I'm not going to go into these. These are just examples of ATP. These all require energy. Exo means going out of this. We're transporting something out of the cell. Endo means something coming into the cell. Do know that these terms, phago and pino, phago means solid. It means cell eating. We eat solids, we drink liquids, right? So pino is some liquid substances being transported. So endo means it's being brought into the cell. Exo means it's leaving the cell. Here's an example of one that requires a receptor to bind before it'll bring it into the cell. Okay, so then moving on, I want to look at the processes of protein synthesis. And before we do that, I want to have you